Hello and welcome to some online class. Friends, today we shall discuss about attenuation. Deletion that remove a part of the tryptophan leader region result in an increase in the expression of the tryptophan operon. However, this deletion will have no effect on the repressibility of the system. That means the tryptophan operon can be repressed as well as de-repressed. This indicates that the biosynthesis of tryptophan biosynthetic enzymes is regulated at a secondary level that require a specific sequence that is present within this leader region. Friends, this diagram represents the tryptophan operon. The tryptophan operon is a cluster of 5 different structural genes along with regulatory region. The second level of regulation is known as attenuation. Attenuation was discovered by Charles Janowski, Kevin Bertrand and his colleagues. The word attenuation is derived from the verb attenuate. Attenuate means to weaken or to impair. The process of attenuation causes premature termination of transcription of tryptophan operon. The specific sequence present within the tryptophan leader sequence that controls the process of attenuation is known as attenuator. The premature termination occurs at a site near the end of the leader sequence. The process of attenuation requires tryptophan charged tRNA. Whenever attenuation occurs, that leads to synthesis of a truncated mRNA. Friends, this figure indicates various components that constitute the tryptophan operon. This is the TRPR gene. The TRPR gene encodes a protein known as repressor protein. After TRPR gene, this is the promoter region. After promoter region, this is the operator region. After operator, there is a specific sequence that is known as leader region. Within this leader region, there is a specific sequence that is known as attenuator. This attenuator sequence plays an essential role in the process of attenuation. After this leader region, there are 5 structural genes that encode various enzymes that are involved in the biosynthesis of tryptophan. The attenuator region contains a nucleotide pair sequence that is essentially identical to the transcription termination signal that are present at the end of the most of the bacterial operon. These termination signals contain a GC-rich palindrome followed by several AT base pairs. The signals are transcribed to form a nascent mRNA that has the potential to form a hairpin structure. When this hairpin structure is formed, it causes a conformational change in the associated RNA polymerase, thereby causing termination of transcription. So friends, this is the leader region. This leader region contains a specific sequence that is known as attenuator. This leader region is transcribed to form a 162 nucleotide nascent mRNA. This is the nascent mRNA, also known as tryptophan leader sequence mRNA. This nascent mRNA begins with an initiation codon that is AUG, and at the end there is a stretch of uracil residue This nascent mRNA also contain two tryptophan codons. These are the two tryptophan codons. In addition to this mRNA contain four different regions. Region 1, region 2, region 3, region 4. These four different regions participate in base pairing to form different types of secondary structure. Already you know that in prokaryote there is no membrane bound nucleus. So the process of transcription is coupled with translation. That means the mRNA can be translated while its synthesis is still going on. So any event that occurs during translation can also influence the process of transcription. In addition to the four different regions, the nascent mRNA also contains a translation termination codon that is present between region 1 and region 2. This translation termination codon is UGA. And already you know that this nascent mRNA also contain two contiguous tryptophan codon. This nascent mRNA also contain a ribosome binding site for initiation of translation at the initiation codon. Available evidences indicate that a leader peptide containing 14 amino acids is synthesized due to this translation. Then we shall discuss about these four different regions. The region 1 include nucleotides from 126 to 134. Nucleotides 75 to 83, it constitutes the region 2. Nucleotides 110 to 121 constitute the region 3, and the region 4 include nucleotides from 126 to 134. So these different regions are present in this nascent mRNA. These four different regions containing such type of nucleotide sequences by which region 1 can base pair with region 2, region 2 can base pair with region 3, region 3 can base pair with region 4. Similarly, region 2 can also base pair with 1 and it can also base pair with 3. But obviously at a time, region 2, it can base pair with either region 1 or with region 3. This indicates that these four different regions can form two alternate secondary structure. In one alternate secondary structure, region 1 base pair with region 2 and region 3 base pair with region 4. Such type of base pairing between region 1 and region 2 and between region 3 and region 4, it leads to the formation of a transcription termination hairpin. This is the structure of the transcription termination hairpin that has been formed due to the base pairing between region 1 and region 2 and also between region 3 and region 4. So such type of base pairing leads to the formation of transcription termination hairpin, which can cause the termination of the transcription. The second type of secondary structure can also be formed, which cannot cause the termination of transcription. In this type of secondary structure, there will be base pairing between region 2 and region 3. When base pairing occurs between region 2 and region 3, in that case, region 3 cannot base pair with region 4. So the transcription termination hairpin cannot be formed. In this case, the process of transcription will proceed as usual. The attenuation of tryptophan operon depend on the cellular concentration of tryptophan charged tRNAs. So friend, while we are discussing about attenuation of tryptophan operon, we have to discuss about two different situations. In one situation, there is low level of tryptophan. When there is low level of tryptophan, there will also be low level of tryptophan charged tRNAs. So the process of attenuation depend on the cellular concentration of tryptophan charged tRNAs. When there is low level of tryptophan, in that case, the tryptophan charged tRNA will also be not available. When the tryptophan charged tRNAs are not available, in that case, the ribosome, while translating the leader mRNA, this ribosome start on the tryptophan codon. These are the two contiguous tryptophan codon. So when there is low level of tryptophan, the ribosome start on this tryptophan codon. So due to this, the formation of hairpin between region 2 and region 3 occur. So the base pairing between region 2 and region 3 prevent base pairing between region 3 and region 4. Due to base pairing between region 2 and region 3, region 3 will not be available to base pair with region 4. So the transcription termination hairpin will not be formed. As a result, the process of transcription will proceed as usual. The other situation is when there is high level of tryptophan. When there is high level of tryptophan, there will be high level of tryptophan charged tRNA also. So when there is high level of tryptophan charged tRNA, in that case, the ribosome move past the tryptophan codon. The ribosome move past the tryptophan codon and it will move into the first and second region. This is the ribosome. So due to this, region 2 will not be available to base pair with region 3. So region 3 can easily base pair with region 4. So friend, here you see there is base pairing between region 3 and region 4. When there is base pairing between region 3 and region 4, this leads to the formation of transcription termination hairpin. As a result, there is termination of transcription. So the attenuation of tryptophan operon ultimately depend on the cellular concentration of tryptophan charged tRNAs. And when the transcription termination hairpin form, it causes conformational change in the associated enzyme, that is RNA polymerase, and cause the termination of transcription. So friends, this is all about attenuation from SAM online class. This is Dr. Subrat Kumar Mohanty. Friend, please subscribe my YouTube channel SAM online class. Thank you very much. See you again. Bye bye.